Before the break, we looked at the process of transcription. That was the first step in the protein synthesis process. In transcription, we saw that we copied the DNA onto a small strand of messenger RNA, and that messenger RNA now moves out into the cytoplasm where the second step, called translation, will happen. Now, translation, you understand what the meaning of that word is. It means to take from one language and rewrite exactly the same meaning in another language. In biological terms, that is exactly what translation is. It takes the nucleic acid language or code and it turns it into a protein code. So it does a translation process. Now, we have a whole lot of nucleotides. Clearly, one nucleotide cannot code for one amino acid because we've got a problem. There are 4 different nucleotides, but 20 different amino acids. So we can't have a one-on-one coding. For example, every time there is an adenine, well, that must be the amino acid valine, for example. It doesn't work that way. You can see that there's a problem with 4 and 20. So how are we going to solve this problem? 4 nitrogenous bases, only 4, and 20 different amino acids. Well, the answer lies in what we call the triplet code. Every 3 sets of nucleotides that we have, every 3 groups of them, we will code for 1 amino acid. So 3 nitrogenous bases codes for 1 amino acid. So a stretch of DNA that originally had 1, 2, 3 nitrogenous bases, we're going to see 1, 2, 3 amino acids produced from that particular strand. And of course, as you know, the DNA is extremely long, so there's going to be an opportunity to have many different amino acids linked together. We need a different kind of RNA to help us with this translation process. And so we meet another kind of RNA called transfer RNA, and we write that with a little t. Remember, you've already met mRNA, which is the messenger RNA, and now we have tRNA. What does tRNA or transfer RNA look like? Well, first of all, the most important thing about the tRNA is that it has an amino acid attached to it. Right up at the top here, at the top end of the tRNA molecule, there's an amino acid attached to it. And every type of tRNA has its own particular amino acid. Now work this out. If I told you there are 20 different amino acids, how many different kinds of tRNA are there going to be? 20. There's an interesting thing about this molecule. It is a single strand of RNA that is folded back on itself. And where it folds back on itself, it bonds complementary, almost in the fashion of DNA. So it's like a paper clip. It curls around, doubles back on itself, and then little bonds are formed to hold that molecule together. This molecule actually has to move around quite a lot. It has to carry an amino acid, which is a large molecule in comparison to it. So it has to be able to be quite robust. And so in doubling back on itself and in creating those little bonds, it makes a much more substantial molecule than simply a long strand would be. And also, very, very important is here at the base of the molecule, we have 3 exposed bases. All of the other bases are linked together to bring about the stability to the molecule. But right at the end of the molecule, sticking out almost like a fork, you have 3 exposed bases. And these are called the anticodon. And they are going to make complementary base pairing with the codon on an mRNA strand. So let's now see what we've got. We've got three actors in this process of translation. We've got the mRNA, which is bringing the message from DNA. We've got the tRNA that's going to bring together the protein monomers, the little amino acids. And we need another structure that's going to work to bring those two types of RNA together to actually translate the DNA code into, or the nucleic acid code into a protein code. Let's have a look how this works. A ribosome engages with or attaches itself to the mRNA, and there is your third actor in this drama, and that is the ribosome. A tRNA will engage the first mRNA codon with its anticodon, not just any tRNA, but one which recognizes and is complementary to the codon with its anticodon. The tRNA will be carrying its specific amino acid. The ribosome then jumps along the mRNA to the next three bases, and a new tRNA carrying its specific amino acid will attach its anticodon to the next mRNA codon. And the first amino acid bonds to the second one. And so the tRNA moves down the mRNA again, and a new amino acid is bonded to this growing chain of amino acids, which is going to make up the protein. You need to be able to be very familiar with this process of protein synthesis, so go and read about it and work through lots of questions from past papers. I look forward to seeing you next week. Goodbye.